All right, thank you guys again for coming. This is Mr. Zawacki, and today we're going to be talking about the scientific method. What the scientific method is, guys, it's very simple. It's just a step-by-step process that scientists tend to go through when they're doing their work. It always starts with asking a question. That's first. Then they're going to do research, look up information, maybe find out the answer that way. If not, they're going to make a hypothesis of what they think the answer is going to be. Then they're going to test it with an experiment. Then they're going to analyze the results with data tables or a graph. And then finally, they're going to make a conclusion explaining how everything worked out and if their hypothesis was supported or rejected. So the first step, number one, you always have to start with a question, always, or a topic, or you got to have something to get started. If you don't have a question or a problem, you can't go anywhere. So that's why it's always first. You want to be able to try and answer your question or solve your problem. That's the main part of it. So we're going to be following this example throughout the live lesson. And the example question is, what color light bulb will a plant grow the tallest under? So we can start thinking, well, there's all different kinds of colors. I wonder which one the plant will grow the best. So second step of the scientific method, once they come up with that question, which is a good question, they need to do background research. Sometimes they might just be able to look it up and they won't even have to test it. Other times, they just get more information. So you already have your question or problem. That was, which light bulb is going to work best with plants. So what do you know about it? Well, plants need light to survive. They do photosynthesis, where they create sugar from light and carbon dioxide. We're going to talk about that, soon. And then write what you know. They look up your question or problem using keywords to find out more information. Do all color light bulbs give off the same amount of light, the same kind of light waves? So the more you know, the better off you can design your experiment. That's what background research is for. That's why it's the second step, to give you more information when you do the lab. Third step is to construct a hypothesis. A hypothesis, guys, we've heard that probably since almost kindergarten or first grade. We know what a hypothesis is. It's a possible answer based on your background knowledge of an educated guess. And generally, and I saw a lot of you guys, I didn't take off any points if you didn't this time, but you do want to start writing a hypothesis with an if, then, because statement. And I know it's a little tricky at first to get that organized in your head in terms of an if, then, because statement, but that's how a hypothesis, at least a formal hypothesis, should be written. So this person came up with, if I put plants under red, blue, green, yellow, and white light bulbs, then, so I got if I put them under those light bulbs, then the plant under the white light bulb will grow the tallest, because the white light bulb imitates the waves from the sun the best. Okay, that's perfect. That's a good way to write a hypothesis. So try your best. I won't take off points in the first lab, but try your best to put a hypothesis in an if, then, because statement. Usually, when they do the if, they add the independent variables here, which we'll talk about what those are in a second. And then they're going to choose what they think is the answer with then. The white light bulb will grow the tallest. And then the because part is usually explaining why they think it's going to grow the tallest. So that's a good way to think about it. All right. So once they have the hypothesis, now you just got to set up the experiment. And in order to set up an experiment, they have to have a really good procedure. It needs to be set up step by step, for sure. Somebody else should be able to follow the directions and do the experiment the same exact way. That's the part of writing a good experiment procedure. The person next doing the experiment should do it exactly the same way you did it. And each step should have its own number as well. So, this is for you guys to participate in the chat box. Is this a good or bad procedure? There's numbered steps, 1 or 2. Gather all your materials, plant all plants, and put them into the bulbs. So that good or bad and why? See, lots of bads, but why is it bad? Good job, Danielle. Yeah, should be more detailed. Asia, good. It's too vague. It doesn't involve half the steps. There's more steps there. It's not descriptive enough. Does not provide enough information. Too basic. Very good. Yep, you guys got it. When you see this, you'd be like, what materials? What plants? What bulbs? How long? You know, so that's why this is a bad procedure. If you're continually asking questions about it, it's not a good procedure. Good job. So we already said why. That is all you guys explained there. So yes, we knew that was a bad procedure and why. What about this one? Take a look at that one. Right. This is a good one for obvious reasons. You know, there's more details to it. There's more steps to it. The same person might be able to copy this experiment after the other. So yeah, it definitely gives enough information. Right. And it doesn't leave a lot of room for questioning either. That's the part of a good procedure. It shouldn't have you question things after reading the procedure. Good job. And we already explained why. It was more detailed. It left out questions that weren't necessary. So good job there. Now, when an experiment is done, this is the important part. If you're going to get anything new or anything that is a review for the day for you, I want you guys to definitely know these parts of an experiment, because you're going to see these guaranteed on your EOC exam. You're going to see these guaranteed on chapter tests. You're going to see them pop up. It's a big part of science. So when experiments are done, they have different variables. And the best way to think about it, for the independent, it's always the biggest difference in the lab that is set up on purpose. So what is changing on purpose? What's the difference? And in this case, it'd be the color of the light bulbs, right? We have a white, a red, a blue, a green. So the different light bulbs, that's the purpose difference. That's the independent variable, whatever is the difference. Dependent, you want to look at D for data. D for data. Because dependent variable is going to be the data behind the experiment. That's the important data that they measure. It's the height of the plants every other day. That's also your dependent variable for your first lab in 101. Giving you guys a couple answers. But they also measure the height of the plants every other day. So dependent variable is that. D for data. Everything else in the lab, there's going to be tons of constants, many different constants. The watt bulb, the soil, the pot, the type of the plant, the amount of water that each plant gets, where they're located, when they are watered. Everything else has to be exactly the same, or you're not going to get accurate results. So constants, there's many, but there's only one independent variable, and there's only one dependent variable. And then sometimes, if there's just two groups, you might see a control group versus an experimental group. The control group is treated exactly like the others, except the independent variable is not applied to it. The experimental group would get the independent variable. So like, for example, if they did a test on a pill to see if it gave people, helped people grow hair, for guys who are balding or something like that. Well, if they gave 100 people the pill and they gave 100 other people a fake pill to see the difference if the pill would work or not, the control group would be the group that got the fake pill. The experimental group would be the group that got the actual pill. So that's the difference between control and experimental. All right. So remember, independent variable is the only difference in the lab. Dependent variable, D for data, that's what you measure. Constants, there's many. That's everything else that has to stay the same. All right, this is the fifth step. So once you are done with an experiment, now you have to do that A word, analyze your data. You should keep track of your data in a chart or a table or a graph. So let's analyze it, guys. Look at this data table. What are you noticing? What are you analyzing? What are you seeing in this data table? Yeah, it tells you the growth. It tells you the growth of the plants. Which one is doing the best, it looks like, after day 9? Couple mixed results here, but let's take a look. Let's analyze it. They all started on day 1. The plants were at different heights, 5, 2, 1, 1/2, 1 1/2. So you can't really say anything there. But you could count the difference from day 1 to day 9. Day 1 to day 9 for the white was 12, 17 minus 5, right? So that would be 12. 2 to 11.5 is only 9.5 millimeters. Should be putting millimeters at the end of each one. Yellow grew 7.5 millimeters. Blue grew 6. Green grew 7.5. So we can see if you guys put white, yeah, white had the best growth over 8 days, for sure. Which one had the worst? Which one's the worst? Yeah, see? So that's what you want to count. You want to count the overall. That's how you analyze it. Look at the overall difference. And blue, yeah, blue only grew 6 millimeters in about a week, a little over a week. Everything else grew more than that. White grew the most, which really surprised you because that does mimic the sun's rays the most. So when we analyze our data, you can put it in a data table, like here, or a graph. And the graph sometimes is even better because you can see easily here, here's the white light, here's the white light graph line. And you can see that one grew the most, and blue grew the least. So that's a good way to analyze it as well. Another good thing to remember, guys, and you may not have known this, but the x-axis and the y-axis are the variables. The x-axis is usually your independent variables, what you're measuring over time. The y-axis is the height of plants. That's your dependent variable. That's a good way to put it into a graph. Dependent variable is usually y-axis, up and down, and then the x-axis is sometimes your independent variable. So we already did this. Which plant grew the tallest? It was white. Which plant grew the least? It was blue. What happened with all the plants? Well, they all grew, so they all had some light, so they all grew some. Just different colors allowed plants to grow taller or quicker than others. So that's what we see there when we're analyzing our data. That's the next to last step of the scientific method. The last step and the sixth step is the conclusion. In the conclusion, the important part is you need to talk about your hypothesis, state whether the hypothesis was correct or incorrect and why, and you should use your data table and analyze, and you should write that in your conclusion as well. The conclusion summarizes all of the results. So that's why it says the plant under the white light bulb grew the tallest, proving my hypothesis correct. See? But it explained why, reaching a height of 17 millimeters, while the plant under the blue light bulb grew the least, only reaching 6.5. The plant under the red was the second tallest at 11.5. It was still much shorter than the plant under the white light bulb. Therefore, plants will grow 147% taller under white and 261% taller under the white versus the blue. So they just went really in depth with explaining their numbers there, which is good. We already know by reading this conclusion that white worked the best, and here's your numbers that prove it. So somebody can do this lab again and hopefully get something similar to that if they follow the right procedures. Okay, so now for you guys to participate, we got a couple questions at the end, and you just guys have to fill out A, B, C, or D, but I'm going to turn this into a poll. So I want you to read the question while I set up the polling. And all you have to do is click the little letter A right below my name, right above you guys' names. Click the little letter A and then choose A, B, C, or D. So look at that question and tell me which one do you think is correct. What's the next best step the scientists should take? So A, B, C, or D. All right. It looks like all of us kind of agree on C, which is correct. So the paleontologist is interested in the size and shape of a recently discovered bone. She thinks that it may have come from a previously known species of dinosaur. Microbiologist has isolated a type of bacteria he thinks may be responsible for a new disease. So what's the next best step? Well, that sounds like a hypothesis to me. So what's the step after a hypothesis? Making an experiment. So that person would design investigations to test their theories. So if you figured out that that's a hypothesis in the question, so what's the next step? Experiment. That's the point of that question. So good job. So it was C. Good work. Oh, another side conversation, but if you guys are interested in dinosaurs, I would really recommend, if you guys get to go out west sometime, go to a place, it's close to the Grand Canyon. It's called Moenave, M-O-E-N-A-V-E, and it's owned by the Navajo Native Americans, actually. And a person will walk you around and show you actual dinosaur bones, fossils, footprints, skull remnants. It's crazy. So if you guys ever get to do that, it's right near the Grand Canyon. So if your parents ever take you there, ask them to go to Moenave as well, because that's a really cool place. All right, so sorry for that. All right, so another question, A, B, C, or D. Scientists once thought there was no life on the deep sea floor due to the lack of sunlight. However, in the 1970s, complete ecosystems were discovered on the sea floor that depend on energy from chemical reactions rather than from sunlight. So which conclusion can be drawn from this discovery? So A, B, C, or D. And yes, Emma, you can text me anytime about that. I'll let you guys know more information. So A, B, C, or D. Which statement can be drawn from that discovery? So it looks like we got mostly C's, a couple D's. So let's eliminate A. We don't have that. Under any conditions, anytime you see that, any, all, everything, be very careful to choose that. And you're right, that's wrong. D, or observations collected on existing organisms can apply to any organism. Remember, don't like answers with any, all, everything. You got to be really careful. So are these either B or C? Observations will always lead to the discovery of new organisms, or organisms may be discovered in remote areas in extreme conditions. Well, we can't say always, remember? So it's not B. It's not, you don't always get a discovery of a new organism. But it is C. Organisms may be discovered in remote areas in extreme conditions. That is a comment that could be true. It's a conclusion that could be said, for sure. And yes, there are actually microorganisms everywhere. There's tons. You don't even know. But lots of microorganisms and even viruses that are mostly harmless, not harmful, harmless, that are around everywhere. So it's just a world that you cannot see because it's so small. And there is life on Mars. People believe now, scientists believe now that there is microorganisms at least on Mars. Not actual beings, but microorganisms, yes. And that's life on Mars. All right. And one more question, maybe two more questions. Okay, so planarians, those are like little worms. They're freshwater organisms that can regenerate. Sonia had three. The Petri dish is like a circular dish. She had three of those dishes with planarians on them. In the same room, the first Petri dish was inside a cabinet, second dish was sitting on a desk, and the third dish was partially covered by a book. So after 1 hour, she noted the distribution of the planarians in each Petri dish. So what was the independent variable? Remember, what's the only difference? What's the independent variable in this experiment? Color, light, movement, or temperature? What do you guys think? A, B, C, or D? Remember, she had three Petri dishes. One was inside a cabinet. That sounds pretty dark. Sitting on her desk. That sounds pretty light. Third one partially covered. That sounds like half and half. So that's the only purpose difference, right? So that's the independent variable. The amount of light is the independent variable. So that's what she was looking for. Do you guys know which one of those three would be the dependent variable? Color, movement, or temperature? What do you think she was trying to measure? Yeah, it'd probably be the movement and some sort of data. Maybe the amount of space they moved, the distance, not really sure. But she's going to measure the movement for the dependent variable. Good job. That was letter B. All right, and one more question for the day. Einstein and Galileo each made many important discoveries in science that changed the scientific world. What do their discoveries suggest about the process of scientific study? So what do their discoveries suggest? A, B, C, or D. Actually, it's F, G, H, or I, but we'll say A is F, B is G, C, and then D will be the letter I. Okay, so A, B, C, or D. So keep looking. A, B, C, or D. Yep. Looks like we are pretty much good here, and you guys are correct. Some discoveries, that word some is a pretty good word. Not all or everything, remember? But some discoveries are possible without the use of sophisticated equipment. That's true even to today. Galileo was in the 1600s. He discovered some planets and some things with astronomy. And he had a very basic telescope that you can get at like Chuck E. Cheese for like 20 tokens today. So, I mean, it's crazy. Einstein discovered things by just looking at different things with his own eyes and making assumptions and things like that with different math. So you don't need the craziest sophisticated equipment. People today are discovering ancient civilizations by just scanning Google Earth. I do that. I catch myself doing that myself. So you don't need sophisticated equipment. People are hiking in the woods out west, and they're discovering relics from the past just by walking around. So you don't need sophisticated equipment to make discoveries. You just have to open your eyes and look around and question things. And that's what this whole scientific method is about. So that's what I hope you guys learned from today, the 6 steps of the scientific method, but why each step exists and the order of which it exists and the variables that go with the experiment. So I hope that helps. And I hope you guys learned something today, or at least reviewed stuff that you already knew about. So keep up the good work. And you guys are all set. So thank you guys for coming.